will be telling us about small world network significance.
This is going to be obviously switching gears. We're going to talk about this graph structure of a neural network. And obviously the future is going to be that at some point we'll get to actually understanding the dynamics that a network like this generates. So before we begin, let me just give you the names of the people who actually did the work. One of them is Jonas, who's going to be here. Jonas worked with VG, Shatakuma, who's in the audience on this project. VG collected lots of the data and Jonas did the computation. And we also collaborate this with Ramon and Lab down at USC. So what is the question here? So the issue is that many of you, I'm sure, heard about this issue, small world networks, that was really exploded in the scene by a seminal paper by Watson Strogatz. And basically what they did was that they asked the following question. Let's take, in a graph theoretical sense, two networks. One which is completely regular, the other one which is random. So imagine that these are nodes, and you have some rule where you connect the nodes. So in the regular network, in this particular example, you connect this node to its immediate first-order neighbors as well as to the second-order nearest neighbors, right? Now, in the random network, you have the same number of nodes and the same number of links as this network, but you randomly connect the nodes, right? Okay. So how do you characterize these two networks? Well, in this case, you can come up with two measures. One of them is, how many steps do I need to take? How many nodes do I have to go through to go from node A to any node B? And then you average that over the network. That would be called the average path length. So in human terms, that's the 6 degrees of separation between me and the present. You know, I know someone, you know someone, you know the present. So the other measure is the linkishness of the network, which is basically asking the question, so if node A is connected to these two nodes, then what is the probability that these two nodes will also be connected to each other? So in human terms, the question is, what is the probability that my friends also know each other? Okay? So it describes how locally connected the network is. The previous measure describes how globally connected the network is, right? So once again, you have the average path length and the average clustering coefficient, as it's called. So Watson Strogatz in this paper, they actually calculated the real L and the real C for the regular networks and the random networks. Now notice something that I should mention before we get to the small world network point. That basically, if you imagine in the regular network, you have a high L because it takes a lot of steps to go from point A to point B, whereas in the random network, the L is relatively low. However, in the regular network, the C is high because the probability, i.e. that the probability that the neighbors are connected is high, whereas in the random network, that's very low. Okay, so this one is high C and high L. This one is low C and low L. The interesting thing is that most of the real networks, and that's what Watson Strogatz showed, are somewhere between the two. And in fact, the amazing thing is that you can generate these so-called small world networks just by taking a very, very few percent of the connections from the regular network and throw them in randomly. Then you will arrive at a point where you still have a high C, just like in regular networks, but you dramatically decrease the L, just like in a random. So what they did was they calculated this for film actors, Harvard of the US, and C. elegans. Now, being a neuroscientist, let's focus, we focus first on this data. So amazingly, in C. elegans, for which the microconnectivity is completely known because there's only like 300 or something neurons, there the L is very, very low, 2.6 steps. So in other words, take any two neurons, it only takes on average less than 3 steps to go from any neuron to any neuron. Now the C happens to be 0.28. Now what I want you to remember is that the C elegans L is 2.6. As you'll see, this is going to be a very interesting number when we consider a real mammalian network, which is what we want to do. So let's now just consider just a little bit of history about this. That after that seminal paper by Watson and Soltesz, there was an explosion of papers where people have measured the actual real networks, in metabolic networks, in the internet, and in various other situations. And there are a lot of attempts at applying all this to understanding, for example, cortical networks. But the issue still is, what is the actual L and C in a real mammalian network? So this is what we try to answer. And we focused on the hippocampus and within that dentate gyrus for the simple reason that probably that's the most well-studied area in the mammalian brain in terms of its microcircuit connectivity. So here is, I don't want to bore you with the details, but let me give you a flavor of what kind of data I'm looking for. Obviously, first of all, we have lots of neurons. Okay? The hippocampus is an elongated structure. It's roughly about 6 millimeters in the rat, and it has within it over 1 million neurons. So we go from the C elegans of 300 neurons to 1 million. And you can ask us the question, well, what could be the L? How many steps do we need to go from any neuron to any neuron? How many steps do we have to involve? So this is what we'll get at in a second. But let me just tell you that we do know a lot about the major cell types that are involved in the dentate. So for example, the granule cells are the so-called principal cells, and there are about 1 million of them. So they constitute about 95% of all the cells, or 90% depending on the numbers. But definitely the majority of the cells are granules. There are various kinds of other excitatory neurons, such as the mossy cells in this part of the dentate that is actually called the hides. So the mossy cells are the cells that project back to the granule cells. Okay? Now, there are various other kinds of interneurons, such as the basket cells, the axonic cells, the mob cells, Ipps cells, etc. We don't have to deal with what they actually do, but the point is that a lot of people have spent basically lifetimes characterizing the exact connectivity between type A to type B, type C, type A. Okay? And we can actually extract that information from the literature. Now, in addition to just, if you want, a relational topography, what is very important is to actually take into consideration that the hippocampus has a three-dimensional structure, so the neuronal connectivity also has a three-dimensional structure. So this, for example, is the axonal distribution of a single basket cell that was filled by Yuri Bujarki's lab in 1997. And we created the axonal distribution. This is the soma, and this is going away from the soma about millimeters this way, millimeters that way. And as you can see, it can be described beautifully with the Gauss. So VG went through the literature with extremely amazing actually to gather this. This is, to my knowledge, an example where a mammalian microcircuit has been analyzed to this extent. And we know, for example, the granule cells, 1 million granule cells. A single granule cell connects to roughly 10 mossy cells, 15 basket cells, etc. Conversely, mossy cells, there are 30,000 of them, and a single mossy cell connects to 32,000 granule cells, and etc., etc., etc. And in each case, we know the axonal distribution as well. In most cases, at least. So, John has designed the calculation methods, and basically we took this, if you want, connectivity matrix, which I showed you in the previous slide, and he went through and used various sampling methods and other methods to come up with it was computationally quite a demanding task. So you can imagine to keep this 1 million times 1 million data set and actually work with it at every step and every calculation step. And here is the first result that I'm going to show you. So the issue is that for a network to be small words, so somewhere between the completely regular and the completely random, what you want to have is that when you go from a random to your network, the L shouldn't change much. However, when you go, the C should dramatically increase. And as you see, this is the random network, the same number of nodes and same number of links as in the real dentate. As you go from here to here, the L doesn't change much. However, the C, the clustering coefficient, the cliquishness, the local connectivity, dramatically increases. So what this means is that indeed the dentate gyrus is a smaller network in the strict definition of the word. Well, an additional point, though, is that I want to point out is that we, in this case, we compare the random to the topographic. And by topographic, I mean that we do take into account that a basket cell at this end of the hippocampus connects to granule cells around it with higher probability than away from it. In other words, we took into account the axonal distribution, not just the relational numbers. You notice that the non-topographic network, when we don't do that, where we just treat it as if it would be just a jumbled up network, the C is much less. So it's less of a small world network than if you introduce the real life topography. However, this is actually the same data but with numbers. This is what I want to show you. If you look at the L of the real dentate gyrus, it's 2.6. So that's pretty amazing if you think about it, that we went from C. elegans with 300 plus neurons, where it took you about less than 2.6 steps to go from any neuron to any neuron. And here we have, in a mammalian neuronal network, exactly the same number. That is a pretty amazing finding, and I don't know exactly how to interpret it, whether it's true for any network in the mammalian brain. It could be an evolutionarily conserved, very important phenomenon. Okay, so in the second set of questions that we addressed, we wanted to understand the following. The dentate gyrus and the hippocampus, in addition to being very important in things like learning and memory, it's also very important in seizures and injury. It is one of the most injury-prone areas. So, specifically, for example, after repeated seizures or head trauma, many of these hilar neurons, such as the Mosty cells and other cells, are lost from the dentate. Now, interestingly, the Mosty cells are the cells that connect to granule cells, I showed you, and they are the ones that connect with their axons, go through the hippocampus and make long-distance connections. They are the ones that they are actually in the mammalian brain the most sensitive neurons to any kind of injury. Which is bad news for head trauma, really, in terms of like short-term memory deficits after the quarterback gets hit in the head, but things of that nature. So we have cell loss, so loss of nodes, and specifically those cells are lost that are the long-distance ones. And in addition to that, the principal cells, the granule cells, make local connections. Okay? So the question is then, what do we expect when we lose the long-distance cells and add the local ones? Where the straightforward prediction was that both C and L should increase. The C should increase because we are adding local connections, that is due to axonal sprouting, and L should increase because we are losing specifically the Mosty cell. Notice that these two things that happen are actually interconnected. When you lose cells, then the presynaptic cells lost their targets, so they are searching for targets and they connect to the surviving ones. The two things are interconnected causally. So here is the result. It is as usual, nature is not going our way, or the way we think it should go. So remember that the prediction was that both C and both L should increase. And after this, so this is basically the degree of sclerosis, by which we mean the degree of cell loss increasing parallel to the degree of sprouting. And as you see, as the degree of sclerosis increases, initially the C, this is on this axis is C, this axis is L, blue is the L. So C, as you see, increases initially and then drops off precipitously. I will explain. So this is the one that we actually expected. This was surprising that it drops off. The other thing that was very, very surprising to us was that instead of getting a very rapid increase in L, we essentially got hardly any increase in L up till more than 60–70% of your mossy cells were lost. And it's interesting because in most epileptic brains, or after head trauma, in fact, you almost never wipe out all of your cells in the islets. You generally have 20–25% remaining, which means that you are still at pretty low L network after head trauma or repeated seizures. And if you think about it from a dynamical sense, if you have your long-distance connections still there, that is actually bad for you, quote-unquote, in terms of seizure propagation. Now, why is that the C goes up and then goes down? It's a somewhat esoteric question, but for those of you who are following this at this late point in the symposium, let me explain. So what we are having here is that the granule cells lost their targets and they're connecting to other granule cells. And initially, when we turn around, it's just adding local connections. Obviously, C will increase as neurosis goes on. And the reason why this doesn't happen is the following. So basically, initially, here is your granule cell, and you ask the question: okay, if you're connected to some postsynaptic targets, normally granule cells don't connect to each other. They only connect to other cells in the network. So when they connect to, let's say, this cell and this cell, what's the probability that they are connected? So as sclerosis increases, then you start to have other granule cells, and then they are connected. They can be connected as well. So you use granule cell, and then it's connected to any of the other cell types, and there is a high probability that the two of them are connected. However, after a while, there are basically these cells are getting lost more and more. Okay, the other cells and the predominant surviving neurons, the granule cells, will be there. So what's going to happen is that although they lost their targets and the granule cells are connecting to the surviving granule cell, there are simply so many granule cells that even if I'm connecting to two other granule cells, the probability that those two will be connecting to exactly each other is very low. So surprisingly, in spite of the fact that you're adding adding local connections, that does not necessarily increase the C. And we can discuss this if you want, but let me just say that the main point, therefore, that L doesn't increase dramatically until you lose all of your cells, and at that point, C happens to be very low. So, I'm not going to go through the details of explanation of excitatory inhibitor network. For those of you who are working on the network, let me just say that the total network obviously can be split into excitatory and inhibitory parts and then separately analyzed. Basically, the inhibitory network alone, without the excitatory network, obviously just losing its cells. So there, what happens is that as you are losing your cells, the L is increasing and the C is decreasing. These things can be fully understood. But let me just conclude, therefore, the following. So first of all, this is, to my knowledge, the first example of an analyzed microcircuit in the mammalian brain, and we can definitely conclude that it is a small world network. And for those of you who are interested, we can discuss this. They are not scale-free. So there are scale-free networks also generated a lot of interest. Basically, scale-free networks are those networks that follow what they call a power law distribution. And again, this is somewhat esoteric point, but our findings did show that there is no evidence of scale-free or power law distribution here. But nevertheless, the important point is that it is definitely a small world network. L appears to us to be evolutionarily conserved, at least in these two examples, the C. elegans and the mammalian dentate, because both of them had an L characteristics that was around 2.6. I also showed you that topography does matter, so the fact that the hippocampus is an elongated structure with local axonal distributions following Gaussians or double Gaussians does matter. And during epileptogenesis, L doesn't increase, almost all of the mossy cells are lost, and C shows a maximum at the lower level of sclerosis. And where we want to go with this in the long run is obviously to expand this line of investigation into two directions. One is to include other parts of the hippocampus and eventually, hopefully, maybe the whole brain. And the other, so if you want to extend it into the spatial dimension, and also extend it temporally, and actually incorporate these findings, the L and C characteristics, into building detailed neuronal network models. Thank you.